To observe the cells and cell organelles of an onion peel under a compound microscope. Apparatus/materials required. Fresh onion bulb, forceps and scalpel, glass slide and cover slip, needle or brush. Droppers, iodine solution, as stain, compound microscope. Theory. The onion epidermal peel is a simple and commonly used material to study plant cell structure under a microscope. Onion cells are plant cells, hence they have a cell wall, cytoplasm, nucleus, and vacuole. Staining with iodine solution makes the nucleus and cell boundaries more visible. The cells are rectangular and arranged in a regular pattern. Principle. The experiment is based on microscopic observation of onion epidermal cells, which helps visualize the basic structural components of plant cells and their organization. Procedure. Take a small piece of onion and carefully peel off a thin transparent epidermal layer using forceps. Place the peel in a drop of water on a clean glass slide to prevent drying. Add 1 to 2 drops of iodine solution to stain the cells, this highlights the nucleus and cell wall. Gently place a cover slip over the peel to avoid air bubbles. Observe the slide under low power, 10 times, and then high power, 40 times, of the compound microscope. Observation. The onion peel shows rectangular, brick-like cells arranged in rows. Each cell has a distinct cell wall, dense cytoplasm, and a prominent nucleus. A large vacuole is seen in the center of the cell. The cell membrane lies just beneath the cell wall. The stain, iodine, enhances visibility of the nucleus and cytoplasm. Result/inference. The onion epidermal peel consists of plant cells with a definite cell wall, nucleus, and large vacuole. This confirms the basic structure of plant cells as observed under a compound microscope. Conclusion. The onion peel provides a clear demonstration of the plant cell structure. Important organelles such as the cell wall, nucleus, cytoplasm, and vacuole can be easily identified, confirming that plant cells have a rigid structure with a well-defined nucleus. Precautions. The peel should be very thin and transparent. Avoid air bubbles while placing the cover slip. Use only a small amount of iodine to avoid overstaining. Handle the microscope carefully and adjust focus slowly.